Hello everyone, now I will discuss the results based on the given article. Firstly, for the differentially expressed genes, the initial step taken by the researcher involved standardizing the microarray data from the three groups of GEOs in order to eliminate any variation caused by batch. From 708 differentially expressed genes, 507 are upregulated genes and 201 are downregulated genes by using GSE 26440 normalized data. This indicates that there is a significant difference between disease and the control group for differentially expressed genes. This figure 1 shows the result of differential gene analysis. Figure 1a is a volcano map, while figure 1b is a heat map of differentially expressed genes. The x-axis of the graph 1a represents the multiple of differential expression, log 2FC, while the y-axis represents the negative log 10 of the false discovery rate, FDR. Within the graph, the blue dot represents 507 genes that were upregulated, and the red dot represents 201 genes that were downregulated. From graph 1b, the sample is represented on the horizontal axis, while the vertical axis represents the different genes. High gene expression is indicated by the color red, while the low gene expression is indicated by the color blue. Next, based on analysis of GO and CAC enrichment, all 708 differentially expressed genes were found to be significantly enriched in GO terms and are related to the immune cells, particularly those involved in the activation and degranulation of neutrophils. Meanwhile, Staphylococcus aureus infection and the hematopoietic cell lineage were found to be significantly enriched in CAC analysis. Figure 2 illustrates the outcomes of GO and CAC enrichment analysis. In Figure 2a, the top 20 enriched GO terms are presented, sorted by the largest number of genes. The horizontal axis represents the count of enriched genes, while the vertical axis displays the name of corresponding GO terms. In Figure 2b, the result shows the enrichment of 20 CAC pathway with the largest number of genes. The horizontal axis represents the number of enriched genes, and the vertical axis indicates the names of respective CAC pathway. For calculation of immune cell, after observing the strong correlation in the GO and CAC results, researchers then investigate the variation in immune response among different groups of samples by analyzing the immune cell composition. The result shows these two groups have a significantly different in the relative proportion of 16 immune cells among 22 types of immune cells. Additionally, the pediatric sepsis group demonstrated a significantly higher proportion of neutrophils compared to the normal group. Based on Figure 3, it shows a schematic diagram of the difference between immune cells. In Graph 3a, the distribution of immune cell types in the 130 samples is depicted. The horizontal axis represents the 130 sample, while the vertical axis displays the percentage of each specific immune cell type. In Graph 3b, the immune cell composition in two distinct groups of samples is presented. The horizontal axis represents the two groups of samples, and the vertical axis represents the percentage of each immune cell type in the respective group. Moving on to the key gene screening and construction of PPI network, a total of 577 genes shows a confidence score of more than 0.4. By utilizing the Cytoscape software alongside the MNC algorithm, scoring of each node in the network can be done, allowing the researchers to rank the top 50 genes based on their scores, starting from the highest to the lowest. The intensity of each node's color was directly proportional to its level of importance within the network. For random forest classification and logistic model construction, by using a set of 50 chosen genes, the researchers constructed a logistic regression model 1 based on the training set GSE 26440. In order to develop a robust and easily interpretable model while minimizing the number of variables, they employed a stepwise regression method. As a result, they identified 5 key genes from the initial set of 50 genes, namely TLR2, MMP9, TLR8, MPO, and CCL5. The p-value for MMP9 and MPO genes were less than 0.05, suggesting that they hold significant value in the model. Conversely, TLR2, TLR8, and CCL5 genes with a p-value more than 0.05 were not incorporated into the construction of Model 3. As a result, the logistic regression model tree constructed by using MMP9 and MPO show no extreme point that would affect the model accuracy. Besides, they also constructed a random forest classification model to further evaluate the importance of these two hub genes. From the result, MMP9 and MPO genes were the top two in the mean decrease genus and mean decrease accuracy, suggesting that these two genes were more important in the random forest model variables. Lastly, for functional validation of the chosen biomarkers, ELISA method has been utilized in verifying the possibility of biomarkers for pediatric sepsis. The level of MMP9 and MPO is inversely proportional with the severity of pediatric sepsis with p-value less than 0.05. That's it for the results part. Thank you.